A disclaimer statement as we're a publicly traded company. Okay, so I'm just going to describe ReNeuron for you very briefly. Apologies for those that are already familiar with the company. There is some brief updates in here, nothing that's market sensitive, but just gives you a quick update over the last few months especially, and more importantly what we're looking forward to achieving over the course of this year, all being well. For those of you that don't know us, hopefully you'll get a reasonable sense for who we are and what we're trying to achieve. So ReNeuron is a clinical stage biotechnology company engaged in exploiting stem cell science and converting stem cell science into therapeutic candidates. So we're developing cell therapies, cell-based therapies, and our primary focus these days is on retinal degenerative diseases at a clinical level. And I'll describe the main program in some detail in a short while. Indeed, our lead program is a cell therapy targeting retinitis pigmentosa, which is an orphan drug treatment. It also has fast-track designation as a clinical candidate. I'll come back to that again in a second. And we have early positive phase 2A clinical data with this program that we're very excited by. We're planning to commence what we hope will be a pivotal clinical study, a pre-market study with this program during the course of next calendar year, if we can continue to garner positive data over the course of the remaining current study that's running, that phase 2A study, during the course of this year. So we have a relatively clear line of sight now in terms of development, clinical development, through to the market for this particular candidate in RP. We're also exploiting exosomes. I'll try and touch on this briefly at the end if there's time. Exosomes are nano-sized vesicles that are involved in intercellular communication, and we can harvest exosome candidates from our own stem cell lines, and those exosomes have utility as a drug delivery vehicle, a novel new vector for delivering complex drugs, or indeed as therapeutic candidates in their own right. As a business, we're well funded now. We recently completed a 17.5 million placing an open offer. Both elements of that funding were oversubscribed back in November last year, and that's given us the runway we need to generate critical phase 2A data during the course of next year, and also preclinical proof of concept data we hope with our exosome program during the course of this year as well. So again, the platforms you see here split into four. The HRPCs, our human retinal progenitor stem cells, are the lead clinical candidate targeting retinitis pigmentosa. We have developed, this is a cryopreserved formulation, so it's these cells are frozen down, which makes them easy to ship and store and then administer when required, having thawed them at the clinical site or the hospital. We have phase 2A data, early data that's positive, as I mentioned. And this program is actually partnered already with Fosun Pharma for the Greater Chinese market, Fosun Pharma being a very substantial China-based pharmaceutical concern, and as many of you know, I'm sure they have interests beyond healthcare in the West as well. Our exosome platform, as I mentioned, we actually derive these exosomes from our neural cell line, our CTX cell line, and we know that these exosomes have the ability to be loaded with molecules of interest, small RNAs, microRNAs, and various proteins, oligonucleotides, and so on. And we also know that they can transport these agents across the blood-brain barrier, which of course is important when targeting central nervous system conditions. So we're working collaboratively with pharma and biotech companies to exploit this platform, as you'll see towards the end if there's time. We're also developing induced pluripotent stem cells, again from our CTX neural cell line. And without getting too scientific about this, induced pluripotent cells are cells that behave like embryonic stem cells. So they have all the utility of embryonic stem cells, meaning they can differentiate into all cell types, all other cell types. But their source material is non-embryonic. And what we're doing is reprogramming our CTX neural cells back into an embryonic stem cell-like state, so that we can then differentiate those iPSCs down into new cell lines targeting things like diabetes, so generating pancreatic islets, for instance, with those iPS cells. So again, another platform, again, using our base cell lines as the starting material. And finally, our CTX cell line, which is the first cell line that we were working with, was originally deployed as a clinical candidate against stroke. That's in phase 2, or was in phase 2 development in our own hands, where we have early positive data, again, at a clinical level. And that's now available for out licensing as well. We're no longer deploying our own internal financial resources into that program. It's available for license. And again, like the HRPC cells, it's already been licensed to Fosun for the Greater Chinese market in stroke as an indication. So a quick word about the pipeline, and again, a little bit of repetition here really. The HRPC cells targeting retinitis pigmentosa in phase 2 development with further phase 2A data expected during the course of this year, leading, all being well, to a pivotal phase 3 or 2B-3 study, however you want to designate it, during the course of next year. We hope with a readout a couple of years later. The exosome and iPSC platforms are at their preclinical stage, of course, working collaboratively with other companies to use our exosomes, for instance, to deliver their drugs. And then finally, the CTX cell line, again, a clinical candidate in stroke disability and available for license elsewhere. For stroke or indeed in other CNS diseases where we have some early data, such as Huntington's disease, for instance. Okay, so just focusing on the HRPC program in retinitis pigmentosa in a little bit more detail. Retinitis pigmentosa is an orphan status disease, by which I mean it's relatively rare as a disease condition. And as it has orphan status, it does confer certain regulatory and market-related advantages if you can get a candidate against that disease through clinical development and onto the market. So orphan diseases are actually quite popular in terms of their licensing potential to biotech and pharma, partly because of the ease of development and getting them to market, and also because, of course, it's a rarer disease, and therefore pricing of those drugs tends to be higher. RP is actually quite a large orphan indication. As you can see here, incidence of 1 in 4,000 in the US and beyond. It's an inherited condition associated with over 100 gene mutations, actually. There's already one marketed treatment, Luxturna, which was approved a year or two ago, targeting the single gene defect RPE65, which only accounts for around sort of 3% or so of the RP population. So what we're looking to do is to deploy a cell-based approach, which is not gene dependent, and thereby confer some benefit to these patients using those cells, essentially to replace lost photoreceptors at the back of the eye, the cells that give us sight. And RP as a disease condition is manifest, as you see here in the panels on the right. It's a gradual loss of peripheral vision, leading ultimately to blindness. It can onset fairly early on in life, in childhood even, and can take a number of years or indeed even decades to progress until blindness. So our HRPC cells, again, just swiftly sort of move over this, but the key points here really are that we've developed this candidate to really get as close as we can to a standardized biopharmaceutical. So in order to be able to make it commercially relevant, we've got to be able to not only generate great clinical data, but also have the means to manufacture and scale up this cell candidate to the requisite quality standards. So we spent many years of development working with the original licensee of this technology, which was Harvard, or the Scape Design Research Institute at Harvard, and other academic collaborators over the years. And we now believe we have an optimized clinical stage candidate that we can take all the way through to market in its cryopreserved formulation. And we believe that we can attract attractive pricing for this if we can get it through to market, given the orphan status nature of this program, both here in Europe and also in the US. And as I mentioned, this program also has fast track designation from the FDA as well. So again, doing everything we can to enable the swiftest possible development of this candidate through to market. So where have we got to? Well, we've done a phase 1 and an early phase 2A study in RP patients. So we treated 12 patients in the phase 1 element, 10 in phase 2A. obviously initially targeting safety, and obviously safety is still a feature in our phase 2A studies. But in the 2A elements, we can start to look at measures of efficacy as well, most notably visual acuity, the ability of patients to read letters on a chart, which is one of the best ways of measuring that. Initially, this study started, or the clinical development, I should say, started at 2 clinical sites in the US, in Boston and Phoenix, and we've now opened the study out to other centers as well, as I'll come to in a second. Surgical technique to get the cells in is retinal, subretinal injection. So again, a well-established surgical procedure that's used with Luxturna, that marketed treatment I mentioned. It's basically a day case procedure, so the patients can come in and typically they'll go home the same day. So relatively straightforward to get those cells in and then follow those patients up. And this technique enables the correct anatomical placement of the cells towards the back of the eye in the subretinal layer, as mentioned. So here's what we're excited about. So as I mentioned, we've seen positive early phase 2A data. And in the interest of time, I'm just snapshotting what we saw at the 12-month follow-up point in the 7 patients that had reached that follow-up point when we last announced an update with this study. So 7 patients have reached the follow-up point, and as you can see, they're listed here in order of their level of response, and there's quite a variation in response, but they all did respond positively, and the mean response of just under 10 letters is definitely something that we're very encouraged by. And what we mean by response on letters is the number of letters that a patient can read on an ETDRS chart, which is similar to an eye chart that you'll see at the opticians. It's not quite the same, but a similar principle. And the important thing here is the mean readout of just under 10 letters, and the untreated eye, because we only treat one eye in each of these patients, you can see the mean there is a worsening against baseline of 2.4 letters. So again, very encouraging data, albeit in a relatively small patient set. And of course, the thing we're working on now is to build this data set out further. So we're now in an extended version of that phase 2A study. We've upped the dose from 1 million to 2 million cells injected per patient. We've enhanced some of the efficacy measures that we're using, really to amplify the signal we've already seen positively in these patients to give us the best shot on goal later on in getting into a pivotal study. And as I mentioned, we've opened the study out now beyond the US into Europe, including in the UK, and we're looking at opening another site in Spain shortly. So the next steps really, it's all about generating more data during the course of this year. We mentioned a month or two ago, last month, that we completed the first cohort in that extended group of 9 patients. So really just to recruit the remaining patients during the course of this year, follow them up for a few months, and then go back to the regulator to move into that pivotal stage next year, all being well. And we will roll out the data in conference, as we have done in the past with this study, as the data comes through. And there are some key ophthalmology conferences that we would typically target during the course of this year. Just a quick word on the retinal stem cell field. So here are some runners and riders that we see who are involved in generating advanced therapeutic treatments, be they cell therapies or gene therapies for RP. So we see ourselves as well placed within this milieu, if you will, and it's all to go for. It's not an overly crowded field, and there is a significant prize commercially to be had. So I'm running a little bit short of time, so I'm just going to finish off very quickly on the exosome program, as I mentioned. So again, without getting too much into the science, exosomes are vesicles secreted by most cell types. They are involved in intracellular communication, as mentioned, but importantly, they can be used to carry important proteins, nucleic acids, and the like, essentially as a novel new drug delivery vector. And that's precisely what we're using our own exosomes to do, working collaboratively with pharma partners here. We have a number of collaborations ongoing. There are a number of advantages we believe our own exosome candidates have in terms of their ease of generation, their ease of characterization, purification, and so on, and indeed their ability to carry key proteins key molecules of interest, I should say, to specific tissues, including the brain. So as I mentioned, we're working with a number of leading collaborative partners here. In the early stages, the key thing here is to generate proof of concept data. And if we can do that, then we would hope that that would lead to license deals with those collaborators that we're working with in due course. So again, it's a race to getting good quality data, preclinical in this stage, during the course of this year. Just a quick word to sum up on value realization. How are we going to realize value in our business? Well, here are some good exemplars of what can be achieved in the field with close comparator companies that are using, in the case of J-Sites, a cell therapy approach for RP, as we are, and in the case of Evox and Kodiak, those are pure play exosome businesses who in relatively early stages have also signed significant out-licensing deals with their own technologies. So there is a lot of commercial value to be had here if we can continue to generate the data that we're targeting. So just to wrap up, we do see significant value creation opportunities for ReNeuron over the coming year, both in our retinal program and with our exosome program as well. And we're well funded to exploit those opportunities to generate value. We're well positioned in terms of the data we've generated already, especially with the retinal program, compared to the peer group, both in terms of clinical development candidates and products already in the market. So we're excited by that. It's clear that there's increasing industry interest, both in ophthalmology in terms of advanced therapeutic approaches and indeed in exosomes as a new paradigm for drug delivery or therapy, as underlined by those deals I showed you. And with that, I'd be happy to take a few questions. Thank you very much.
I will get straight on to the questions. The first one I've got here for you is, how are the stem cells harvested, and what, if any, is the cost of this?
Well, the advantages we have with our retinal program are that we harvested the cells, so they are multipotent cells. They're originally fetal derived, and the technology I mentioned that we licensed from Harvard way back in 2007, 2008, enables us to expand from a single sample to expand those cells out to a very large population of cells using low oxygen culture. So essentially mimicking the oxygen environment you get at the back of the eye, which is usually where these cells reside. And in doing that, we can generate very high quantities of cells to the right standards and then just draw on those banks as we need to without having to go back and rederive from fresh tissue samples in high quantity. So again, it's part of the allogeneic approach, the non-patient specific approach to cell therapy. Can you generate high cell populations or cell lines so that you don't need to continually source from fresh tissue?
Okay, thank you. Got a question here from a shareholder. Pleased to participate in the latest fundraise through the open offer to buy new shares at a cheap price. Disappointed though, the number of shares I could apply for was restricted, whilst major shareholders could apply for an unrestricted number. Will the company make its next fundraise open offer more open to small shareholders?
Yes, I mean, I think if we're very honest about that, we did not anticipate the level of demand. I mean, it's clear that the raise was priced to go, as they say, and I have to say, based on prior experience, we did not expect to get an oversubscribed open offer. And I think if we had our time again, we would have done just as that shareholder has recommended. It's probably not quite right to say that the placing element was unrestricted. Clearly, we were pegged back in terms of how many shares we could place into the market, be it through the placing or the open offer. It's never as open-ended as that. We did scale back institutions at the end of the day as well. But I think it was the open offer certainly that surprised me in terms of its demand, and it's a very fair comment to make. And I think next time we would look to improve that ratio to offer more shares to directly to existing retail holders.
Okay, perhaps that leads me on nicely to the next question. There's one particular analyst offering a 12-month price target, expecting the share price to move to 400 pence in the next year from the latest price of 105. Is there any comment to make on that?
Well, we have a number of analysts that typically follow us, and some are more aggressive in their pricing estimates or projections rather than others. I don't think we particularly make any comments on their reports. I mean, I think the analysts, do their own work and value the company on a number of different bases. I think for us, if we can continue to, or from our perspective, if we can continue to garner the data that we've seen so far in the retinal study, which is extremely encouraging in its early stages, then we would certainly accord with the kind of valuations that those analysts are attributing to the business respectively. Why wouldn't we? And there's plenty of precedent as well in the field for value generation, as I touched on in the presentation, and beyond that, actually, in terms of companies that have been acquired in similar fields once they've generated phase 2 data that's really convincing. So I think there's real value to be had, but this is biotech. It's incredibly difficult to predict, and when it does happen, it can happen very, very quickly. But there's clearly risk attached to that equation as well.
Absolutely, absolutely. Okay, why choose a licensing partner from China rather than closer to home?
Yeah, it's an interesting one. I think the simple fact of the matter is we saw increasing interest from China in the early stages of those programs. We weren't actively chasing licensing deals either in the East or the West, but we were approached actually by a number of potential partners in that region, and China specifically. And Fosun weren't the only suitor, if you like. And we chose Fosun because they are one of the leading players, and they do have precedent actually in terms of licensing advanced therapies, gene therapy in particular, actually from the West. So that gave us some confidence that, if you like, they knew what they were doing on the ground in China, and we're absolutely delighted to have them as a partner. We couldn't hope for a better one in that territory. But I do take the point. It looks a bit odd doing a deal in the East as a Western company, but there we go. A deal's a deal.
Yeah, that's right. Okay. Will the allogenic cell-based therapy for RP activate an immune response, and how will this be managed?
Yeah, that's a good question. I mean, typically the cell lines we're working with, both the retinal line and the neural line, don't engender immune responses. So we don't have to immunosuppress patients in this RP study, and indeed we didn't have to in the study that we were running with the neural cell line in stroke. So the cell lines or populations were chosen partly on the basis of those particular characteristics. Now, I think where it becomes more relevant is if we choose to repeat dose, which could be on the cards for treatment for RP. At the moment, we need to generate more data to show that repeat dosing is going to be safe and effective, because it's the repeat dose that might ultimately amplify an immune response in these patients, as you might expect. But certainly on a single one-off shot, which is ultimately what this therapy is envisaged to be when it's administered in market, we don't see an immune response, and we never have done, and we don't immunosuppress the patients on that basis.
Okay, got time for one more question. Please bear with me, quite long. If large therapeutic molecules, for example, monoclonal antibodies, could be transported across the blood-brain barrier, this could give rise to have multiple new exciting applications. Do you anticipate that ReNeuron's EXOSOMES will be suitable for transporting monoclonal antibodies across the blood-brain barrier, as well as smaller molecules?
Yeah, it's there is probably a size limit, and again, it's probably beyond my pay grade scientifically to answer that question with any great authority, although as happened last week, if you have a more technical question, please do write in at info@reneuron.com, and we'll try our best to answer them at a more technical level. But that's exactly what we're testing at the moment. So we are testing a whole range of molecules, both large and small, to see what we can do, both in terms of directing them across the BBB and also directing them to specific tissues. And we can actually engineer our exosomes into different types of exosome candidates so that they are better directed at specific tissues. So we can actually adapt individual exosome candidates to do that, dependent on what they're carrying. But it's fair to say it's in its early stages, and I don't think we have a sufficient enough data set yet to be able to answer convincingly, at least at a preclinical level, what these exosomes can and can't do in terms of transportation. We have some early data, but it's speculative, I think, at this stage, and that's precisely what these early stage collaborations will build on during the course of this year and beyond.
Excellent. Okay. Thank you very much, Michael, for the presentation and answering the questions. I appreciate if I didn't I'm sorry if I didn't ask all the questions being asked this evening, but we've run out of time now. Thank you very much.